So it's my pleasure to introduce Darren Monckton, leader in the repeat expansion field. He's also on our advisory board and just one of those all-around great guys. So if you need some mentoring or advice, Darren is your guy. So later today and tomorrow, find Darren.
Well, thank you, Andy, for that very generous introduction. And again, absolutely happy to talk to anybody about any aspect of myotonic dystrophy or any of the other repeat expansion disorders. And again, really thanks to MDF for organizing this meeting and giving me the opportunity to talk to you today about some of the challenges that we face in myotonic dystrophy in terms of the variability of the condition, but also the opportunities that it also provides. And again, thinking a little bit about where we are currently at in terms of very exciting time in developing and having active clinical trials ongoing at the moment, and looking forward, obviously, to the stage at which there are approved therapies, and very likely will be potentially within a few years, multiple approved therapies. I think one thing we need to consider is that probably, at least at this stage, none of those are going to be curative treatments. They're hopefully going to be effective in a large proportion of the population. But again, one thing we're going to have to do is understand the relative efficacy of individual drugs so that we can compare and ensure that any one individual is on the best kind of combination or individual drugs or combination of drugs for that individual. So really working towards this idea of having personalized medicine. So next up is going to be my disclosures. They will appear and I will go through them very quickly, or I'll just zoom over them. So I have disclosures, okay? So I have various relationships that you guys should be aware of, academic and scientific. Okay, so this picture here from Rick Giordotti is really nice. I think it really captures the diversity of myotonic dystrophy in the real world. So obviously we know that DM affects individuals of both sexes and all ages, and affects a wide range of different systems, and that every one of those individuals is having their own absolutely unique experience of DM1, different onset, different constellation of symptoms. And that obviously complicates things, but it also provides us with some opportunities. If we could understand what is driving that variability, then it will give us insights into the disease process. And obviously, as a human geneticist, one of our primary hypotheses is that a good chunk of that variability is genetically driven. And obviously, with a genetically driven process, we have absolute ways to try and understand what those genetic drivers are. So that phenotypic variability complicates on multiple levels. So obviously, it impedes our ability to provide accurate prognostic information to individuals and families about the course of their condition. It obviously complicates our ability to define what is the natural history of myotonic dystrophy, because again, as I've alluded to, there is no one version of this condition. Everybody has their own unique version of the disease. And again, that obviously then when we're thinking about clinical trials and developing biomarkers, again, that is complicated by this extreme variability in the population. And again, then when we want to figure out whether a drug is working, again, each person in the trial will have started from a different position, will be progressing at a different rate. So that means then checking out whether the drug has worked becomes more complicated. And then again, as we go forward, understanding how individuals are responding to the drug, again, is going to be really important for ensuring that we have the optimum treatment strategy for any given individual. So until we can understand that variability, we're not going to be able to drive and in many of these areas as efficiently as we could do. So if we understood better what was driving variability, we'd have greater insights into the disease course, more accurate prognostic information could be offered to families and individuals. We'd be better able to delineate what are the most effective biomarkers and outcome measures in clinical trials. That would lead to more efficient clinical trials, and we'd reduce the risk that we might miss a particularly effective therapy, or even worse, in a way, think that a therapy is effective just because of the variability in the population. And again, going forward, better understand individual drug responses. And again, also by understanding the biology, we might identify new targets for therapeutic intervention. And we've heard over the last couple of days, the idea of precision medicine. We, again, are a long way from being able to deliver the precision medicine in myotonic dystrophy until we understand much more about this variability. So, fortunately, we're not working in a complete vacuum. We already know that the number of repeats inherited is the primary driver of disease severity in this condition. That individuals who inherit a large number of repeats have a much earlier age of onset, and those who inherit a relatively small expansion, 50 to 100 repeats, for instance, have a much later age of onset. And we can see in this data here, generated by Fernando Morales from the Costa Rican cohort, which is a family-based study. So it includes individuals with the very late mild onset form of the disease and children with the severe juvenile and congenital forms of the disease, that the inherited number of repeats explains about 70% of the variation. But that's really driven by those two extremes. If you look in the middle there, but from 2 to 500 repeats, that there's much more variation that we don't really understand yet. And that again, is particularly the window that are most likely to be involved in the early clinical trials. So we understand less about what's driving that type of variation. That variation, however, as we know, is not randomly distributed within and between families, follows a highly reproducible course of this anticipation that we've known about for a long time, where the disease frequently becomes earlier and more severe in successive generations. And that's because the repeat expands from one generation to the next. And in myotonic dystrophy, this is particularly dramatic. So the repeat typically expands by hundreds of repeats per generation, such that in a very typical myotonic dystrophy family, we go from a mild late onset form of the disease in the first generation to a severe juvenile or congenital form of the disease in the third generation. So that's an anticipation of 20 to 30 years per generation. And what that often means is that those earlier, milder forms of the disease are more difficult to diagnose. So many families are only diagnosed when a severely affected child is born. And what that means is we have massive ascertainment bias within the myotonic dystrophy families, that we mostly only find about the milder forms of the disease once someone with a severe form of the disease has been diagnosed. So again, we find about those children with the large repeats when they're early, the adult onset generation when they're middle-aged, and then the milder form only when they're already relatively aged individuals. So I'm showing you this graph here. You're saying, oh, this is the graph I just showed you. Actually, this is a different graph. Now on the y-axis, we have age at sampling. So this is the time at which the people came to the attention of the research community. So again, that's highly predicted by the number of repeats. We sample people with large repeats early, sample individuals with smaller repeats much later. And again, this is the single most significant and highly powered correlation that you're going to probably see during the entire 3 days here. And it's the association between the age at onset of the condition and the age at which those people become into the research community. So this is purely an artifact of anticipation and ascertainment bias. So this is something we need to consider in any aspect of the research that we are doing is impacted by this ascertainment bias effect. So I'm just going to illustrate that here with some other data. So again, repeat length, inherited repeat length is the primary driver of all of the progressive phenotypes. And there's some really nice data here generated by the Canadian group at the University de Sherbrooke, Cynthia Gagnon, Jean Mattel, et cetera, that group. Really generated some nice data on pinch strength here. Again, highly predicted by the number of repeats inherited. Clearly, this is a progressive condition. Pinch strength decreases with age as it decreases with CAG length. But if we just look at the age effect here, in a cross-sectional study, there is no age effect. And in fact, actually, that line goes up a little bit. It's not statistically significant, but that line looks like older people have higher pinch strength. And they do in this cross-sectional study, but only because of that ascertainment bias. Obviously, the lived experience for any one individual is that their pinch strength decreases with age, but actually in a cross-sectional study, it looks like it goes up with age. And again, that is totally an artifact of this ascertainment bias. So if we're going to understand these progressive phenotypes, we absolutely need to include both the CTG length and the age of the individual if we're going to stand any sort of chance of being able to account for that ascertainment bias. Okay. So again, when we add in an age-CAG interaction, then we get the expected relationship here that individuals with larger repeats lose pinch strength at a greater rate as they age than individuals with smaller alleles. Okay, so that massive ascertainment, again, as well as having a huge impact on the families and their lived experience, is really important in any aspect of research into DM1. So if you're going to take home anything from this session, please take home that message. The complicating factor here is that the repeat is not only unstable from one generation to the next, it's also unstable throughout the lifetime of the individual. So what I'm showing you here is some data that was generated by our group as part of the Optimistic project, where we were looking at the number of repeats in blood DNA. For this particular study, we were able to get a DNA sample at the time they joined the particular Optimistic study, but also we were able to access, for some of these patients, the DNA sample that was taken when they were first diagnosed. And often that was 5, 10 or years previously to that. And what we're doing here is we're using small pool PCR. So we're doing essentially PCR with relatively small amounts of DNA such that we can detect the product from individual input molecules. So each one of those discrete bands you see there is the DNA that was present in an individual blood cell. And we've got data here from 3 patients sampled at 2 different age points and multiple PCR reactions for each individual. So you see the person on the left there sampled first at 46, and then when they were 55 years old, they inherited a relatively small allele, probably around about 80 repeats. And you'll see that, again, that there are some alleles there that are going over 100, but the degree of somatic expansion is relatively modest. Whereas in the middle, you see someone there who probably inherited around about 150 repeats. And you see already in the age of 26, there's a wide degree of variation, some alleles up to 200 or 300 repeats. But then when we come back at 48, there are now alleles up to 500, 600 repeats. And again, the individual on the far right probably inherited maybe 250 repeats. And again, that's expanding even more rapidly if we consider how young that person is, 16 and 23, when they were sampled. So what we can assume from these data is that lower boundary probably is our best estimate for what was the number of repeats that was inherited by that individual. It's that number of repeats that's inherited is the primary driver. We can also measure the modal allele. That's the average number of repeats at the time the sample was taken. But what you'll notice is clearly that modal allele is increasing with time. So the average number of repeats in blood is increasing with time. And that obviously comes back to be important then in terms of what actually is the primary determinant of disease severity is the number of inherited repeats plus how quickly it gets bigger. So ideally, we'd be able to get both of those numbers for any one individual at any given time. So what is the diagnostic test? What is which of that information do we actually get from the diagnostic test? As Paul very nicely showed us, the traditional test is southern blot hybridization of genomic DNA digested with a restriction enzyme. And you get these blots here, and where the expansion presents as this fuzzy band from which it's only really possible to detect the average size. So in the best case scenario, all we're detecting is the average size, which, as we know, is changing with time. So this does not give us the inherited number of repeats. Moreover, these are large DNA fragments, and literally 1 or 2 millimeters difference on how far that band runs on the gel is the difference of hundreds of repeats. So this is highly imprecise, and it's only giving us the average number of repeats. And that's the best case scenario. Actually, what is most people are getting, as Paul has alluded to, is just this repeat primed PCR assay. And all that tells us is, are there more than 50 repeats? It gives us no indication of the size at any time point. So clearly the diagnostic test is not going to be particularly useful in terms of trying to understand the variability in the population. However, it's not completely useless. This shows some data here from the Costa Rica cohort, where again, there clearly is value in that number when you get it, at least from the southern blot approach. But again, obviously from the yes/no, obviously contains no information. Okay, so whereas again, if we use that inherited number, which we don't get from the standard test, then again, we can improve things dramatically. That somatic mosaicism obviously is present in blood. Obviously, it's also present in other tissues. And again, you see some data there. I think if Charles is in the room, that I think that is probably Charles Thornton's first paper on myotonic dystrophy, where he very nicely showed that the number of repeats that were present in muscle cells was much larger than was present in the blood cells. So patients presenting in blood with 100, 200, 300 or 400 repeats in muscle cells often have many thousands of repeats. And again, that's data that we and others have replicated. Again, the Costa Rica group have shown nicely that the repeat expansion in skin and muscle is much larger than it is in blood DNA. And that, again, we think is really important in terms of the disease process. Think about the CTG inherited expanding over time in a process that's driven by the number of repeats inherited and is dependent on age and cell type, such that we end up with a much larger toxic CUG repeat in the affected cells than the actual number of repeats inherited. And that's what's really driving the disease process. And again, that has multiple important implications. So the first one is, despite what we heard yesterday about the CUG being the proximal cause of the disease, the CUG is not the proximal cause of the disease. It's an important part of the pathway, and addressing that as a toxic molecule is absolutely important, and we absolutely expect it to be therapeutically beneficial, but it is not the proximal cause of the disease. The inherited CTG is the proximal cause, and there's lots of really interesting biology and important biology happening between the inherited CTG and the toxic CUG. And again, that provides opportunities in terms of additional routes at which we can potentially intervene therapeutically. And obviously, again, that's important probably in the tissue specificity, why in many cases this is a late onset disease, because you need time to acquire those repeats. And again, is also important part of why the disease is progressive. And again, this is ongoing throughout the life of the individual. So again, in terms of any therapies that are being developed now, those therapies will have a given effect at a given time point, but the underlying problem is getting worse with time. So any drug will have to work harder to mediate the same effect as the patient ages. So again, I think that's an important consideration. And then just one other quick thing again is that the number of repeats in blood does not necessarily indicate the number of repeats in a given tissue. And again, when people are taking samples, biopsies, looking for biomarkers, using them as cell models, etc., you really need to know what is the repeat length in those tissues if you really want to understand what is the features that are important about that particular model. Okay, so then one other thing again is this somatic mosaicism repeat length. We also know that in a subset of individuals, there are these variant repeats and probably present in less than 5% of individuals, mostly CCG interruptions. Now, I say they're present in less than 5% of individuals, and they appear to mainly occur as de novo events. So in most individuals, there's a pure CTG, and that increases from one generation to the next using that under that classic kind of anticipation that we see. What occasionally happens is that we get these de novo events where we gain variant repeats. And what happens is when we have those variant repeats, the repeat is relatively stabilized in the germline, so that often in the next generation, it either stays the same, sometimes gets a bit bigger, but actually quite often gets a little bit smaller as well. So what we end up with is those variant repeats tend to be clustered in those individuals with a few hundred repeats. So we don't see them in those mildly affected individuals that are in the first few generations, and we don't see them in those later generations where the repeats are really big. So although overall these variant repeats are relatively rare, they are enriched in that middle group of individuals that are the most likely clinical trial cohorts. Illustrated here in the optimistic cohort, where about 8.5% of the individuals had these variant repeats. And similarly, in the Canadian cohort, again, enriched for adults with the condition, again, we got 8.9%. So these variant repeats are highly enriched in clinical trial cohorts. Why is that important? Again, these variant repeats reduce the rate of somatic expansion, and associated with that is a later onset of the disease. So these individuals with variant repeats typically have an onset 10 to 15 years later than we would have expected if they inherited a pure number of repeats. Same applies to these progressive phenotypes here. Quite clearly, those individuals with the variant repeats are retaining pinch strength longer than those individuals without variant repeats. So it's having an impact on the types of measurements that are being used in clinical trials. So again, we can add another factor here to these variant repeats, modulating somatic expansion and disease course. So what do we know then? That the inherited repeat is an important driver of the disease progress. Anticipation yields this ascertainment bias that is really messing up our ability to really understand the variability in the disease course in individuals. particularly from cross-sectional studies. The allo length is particularly expanded in the diseased tissues, and that has implications for cell and animal models and how we interpret those, and that the somatic expansion is really an important part of the disease process, and that these variant repeats also modulate that on top of both the somatic expansion. What I think we should really be thinking about is, in terms of clinical trial cohorts, is that we should really be ensuring that we're consenting to do more detailed genetic analyses, including potentially whole genome sequencing, that we get to collect biosamples to enable that, and that really we should be, again, not just relying on the simple diagnostic test. We should really be incorporating more sophisticated methods into this process. Again, with the idea that we can, at least at this stage, use these in post-hoc analyses. Now, maybe it won't be important, but again, I'm worried, particularly when the studies are small at this point. Particularly, we have a couple of those individuals who are genetically extreme, variant repeats, for instance. They get in the treatment group and it looks like you are getting a bigger effect than you really have, or they're in the control group and you miss a positive effect because they were going to be more mildly affected to start off with. So I really think we should really be trying to incorporate these into the studies going forward. And I just want to spend the last couple of minutes just again thinking about not just the repeat sequence, but about the rest of the genome. So clearly, cis-acting modifiers, variant repeats, the length of the repeat is important in DM, but at least part of what's driving that variability between individuals is the rest of the genome. Again, we know from other disorders such as Huntington's disease that if we can understand what are the genetic modifiers, then that will tell us something about the disease process. So these are some data from the HD Enroll cohort that have been incorporated into genetic modifier studies in HD, where we're looking for what other genetic variants modify the age of onset of HD other than just the CAG repeat. And what that revealed was that there were other sequence variants in the repeat itself that modify disease severity. Then a whole bunch of DNA repair genes, genes that we know from animal and cell models, are the very genes that are driving somatic expansion. So this really revolutionized the understanding of the disease process in HD. So again, there's clearly work going on to lower HTT, just as there is to lower DMPK, but there is a massive effort now, multiple academic and commercial entities developing treatments targeting these DNA repair genes as being revealed as key modifiers of the disease process. So again, these are data from humans. Essentially, what this is telling us is that nature already did the study. Nature already showed us that modifying the amount of MSH3 modifies disease severity. So this is the best data, preclinical data you can generate, is nature already did your drug trial for you using natural genetic variants. We need this information in DM1. And again, there are other genes in there as well that are not DNA repair genes, but also they are not the pathways that everybody thought were important in HD before these types of studies were done. And again, this is similar data now, just looking at genetic instability as a molecular phenotype. Again, we really need this data from DM1. Again, we have some limited data. Our group has shown that MSH3 variants in that gene, the same ones that are modifying rate of somatic expansion and disease progression in HD, are also modifying the same process in DM1. And that's very encouraging. The idea that we might have a drug that inhibits MSH3 could be affected in both disorders. And again, there's some evidence that variants in MBNL1 might also modify the disease process. And again, we would absolutely expect a variant in MBNL1 to potentially modulate disease response when, again, we know that MBNL1 release is the primary target of the DMPK lowering therapy. So we really need to move to a scenario where we can generate that data and hopefully put a lot more flesh on this type of diagram. So then just finally, we really want to realize precision medicine in DM1, then we have a bunch of challenges ahead of us. So one of them is relatively short to medium term aim of incorporating better genetic testing into the trials that are ongoing. I think that will be even more important as we go forward into phase 4 trials, really trying to understand drug response, so how that differs between individuals. And again, to really exploit that idea about finding more genetic modifiers. Again, we read large characterize cohorts with biosamples available. And Guillaume, again, made that case already today in terms of talking about the DM1 scope registry. That's exactly the type of registry we need to expand and, again, coordinate with better genetic information for all of those individuals. And again, how we do that in large scale, again, is a challenge. The methods I've shown you are based in our lab, again, are relatively low throughput. We and others have made certain advances in using long-read sequencing technology, but again, we're still a long way from being able to apply that in the type of depth to really allow us to take account of mosaicism and really provide the genetic information that we need. Obviously, DM-CRN, again, is potentially getting us in that direction as well as DM-Scope, and the remote assessment procedure that Johanna Hamel is leading from Rochester, again, is another way that we can potentially generate larger cohorts with biosamples and more clinical data available. So this is the challenge for the community going forward, is we need to address these challenges. If we really believe that we're going to be applying precision medicine in DM, then we need to be able to address these questions. And again, we are not unique in this scenario, and there is precedent. I've already alluded to the fact that the major progress we've been able to make in HD is based on the Enroll HD platform that will now have over 27,000 individuals in it at its next release, expected in a few weeks' time. And again, we're going to be very fortunate. In the next few months, we will get 20,000 whole genome sequences from HD patients. This, again, is going to allow us to take us to the next level of genetic analysis. That has to be the aspiration for the DM community. And again, we also heard a little bit about yesterday about the work that Vertex have done in developing treatments for cystic fibrosis. That is the absolute paradigm for precision medicine in the current era. That work is facilitated by the database that the Cystic Fibrosis Foundation organize and fund, that currently contains information on 122,000 individuals with cystic fibrosis. Cystic fibrosis is no more prevalent than myotonic dystrophy. We, as a community, that has to be our goal if we really want to understand and cure this disease. Quickly thank, obviously, all of the families and individuals that have donated samples, my group, and all of our collaborators that have allowed this. And again, we need to have even more collaborators, even more people on this slide. Again, we're going to require even more funders to pay for that. Thank you.
We'll do one question, and then Darren will do a bunch too.
Okay.
Thank you. Ferris from Vertex. Thanks for giving a shout out to Vertex. Great company. A quick question. You really bring parallels in HD and DM1, which I think they're very elegant. And we're learning more and more to go on the upstream to try to really prevent that triplicates repeats that seems to be driving the disease. In DM1, many people are looking at the splicing. Many people have looked in HD at mutant Huntington and haven't led to any therapeutic difference in patients. Do you think we're chasing the wrong target when we have splicing, and we should be looking more upstream?
So I think, again, well, I have to talk about DM1 first of all. So the idea, where should we intervene in that pathway from the lesion at the DNA all the way through to disease? And I think there are therapeutic opportunities all the way through. But I think the general point is that the further back towards the proximal cause of the DNA of the disease, which ultimately is the DNA, the more things downstream we will potentially treat. So in DM1, I think it's quite clear the CUG is not proximal, but it's nonetheless a major nexus of downstream pathogenesis. And I think the cellular animal model data that's been generated are consistent with that. And again, the preliminary data that's coming out of the clinical trials is consistent with the fact that you can have meaningful clinical impact at that particular process. Okay. That's not to say that something upstream might be potentially even more impactful. Again, depending on when it's delivered, I think it's very credible that we can end up in a scenario where we have drugs that are maybe slowing the rate of progression in combination with reducing downstream toxicity from a CUG. So I think we should not put all of our eggs in one basket, and we should spread them out as widely as we can, including as far upstream as possible. For HD, I think, again, there is clearly a major effort on HTT lowering. Animal model data suggests that might be useful. But again, I think real world experience will pan out there. Again, we know there have been some unsuccessful HTT lowering trials. And again, it's unclear how much of that was the fact of lowering HTT, again, non-allele specifically, or whether there was some kind of adverse response just to high concentrations of oligonucleotides independent of their target, I think is to be determined. But again, in there, I think we're again assuming that the toxic polyglutamine is the real intermediate. Again, in cell and animal models, that kind of seems consistent. But I think our recent understanding of the relative repeat size in affected neurons, being probably hundreds, if not thousands of repeats that's really necessary to generate the symptoms, may change our view of what is actually the toxic element in HD, but to be determined. Thank you.